Alright, so section 3 of the research then is mutations and how we use mutations in agriculture. Now, I don't want to sound too much like a history teacher here, but if you go way back to the dawn of civilization, the rise of what we consider to be modern human civilization also came with the rise of human agriculture. That is, farming communities, as opposed to the previous hunter gatherer stages. So we don't have a bunch of clear records on this, but we're pretty sure that humans recognized that offspring took on the traits of their parents before people started building these farming communities. And then when they decided to build a community around agriculture, they knew that they had to grow food, animals, plants, and whatever else, and they had to decide what food to grow based on the traits of the parents that they used to make the offspring. There's a common example is that most lazy teens can understand. If you have ever seen a lawn that has been neglected and overgrown, not just weeks, but sometimes even months, what you'll notice is at the top of the grass you get these little seed pods. This is in fact where if you are going to actually get grass seed to plant, this is where it comes from the top of the grass here. Now, thousands and thousands of years ago, people saw this and said, hey, there's a potential food source. Before you make the decision to feed your community on your overgrown lot, you have to first identify, well, what kind of grass seed can I actually eat? Which ones can't I eat? Which ones should I cultivate and which ones should I avoid? So I introduce you to one of the prime examples of this grass called teosinte. This was a grass that is native to Central America. It would grow to be about 1.5 feet, 2 feet tall, and it had these seed pods at the top. And people would take these seed pods and they would look something like this. You could grind those seeds up and then you could make all sorts of different types of foods with it. One could easily argue that all the great civilizations of Central America were based on the cultivation of this teosinte. One could even argue that most of civilization today, essentially, especially here in the Americas, is built on teosinte. Now, most of you wouldn't recognize this ancient grain of grass, even though that you see it every day, and many of you probably eat parts of it every day. What the humans did here was they engaged a common practice known as selective breeding. That is, choosing which specimens to breed to have offspring. They're able to choose the traits that they want. So, which of these seed pods had larger kernels? Which of these seed pods had more caloric content? Which ones were easiest to grow? Which ones were most drought resistant? And they would choose them year after year, season after season. And what we end up with now is a grain that many of us are very familiar with, known as maize, or to some people, corn. For good or for bad, corn is in pretty much everything we interact with nowadays. There's usually corn in your pancakes, if not you put it on the pancakes. It's in almost every item that you get at the store, the grocery store. We make fuel out of it, we make plastics out of it. All sorts of stuff is made from corn. And all that was brought about by this ancient grain, teosinte. We've seen this type of thing happen again and again. With chickens, we've taken these wild field grouses and turned them into these giant meat birds. And you can't help but feel bad what happened to wolves. In the end, selective breeding has been a very useful tool for civilization. It's what's fed us. It's what we've used as resources for years and years and years. It is a technique we continue to use today to make all sorts of products, all sorts of food products and other things that we grow by choosing the traits of the parents for the offspring that we want to see. While modern farming may have been built on crossbreeding or selective breeding, which one you call it depends on which book you're looking at or which website you go to. Modern farming was built on this selective breeding, but that's not where we stopped. We have developed several other techniques over the years that have also been employed using trickery and heredity. So it was in the early 50s, I want to say 53, that we discovered the structure of DNA. But long before then, we already knew that there was chemistry inside of a cell that determined the outcome of the offspring. And we started kind of trying to play around with that using radiation. So what we're talking about here is something called mutagenesis. The basic idea of mutagenesis is you take living cells, usually the seed cells of an organism, and you would expose them to some sort of mutagen, whether it's radiation or chemicals. Sometimes they use some other methods. Basically, you were causing mutations in the DNA of those seeds and then growing them to see what happened with the offspring. If you recall my bad mechanic example earlier in this video, we had mentioned that if you go in and start changing parts of a car and not knowing what you're doing, what's likely going to be the outcome? More often than not, you're not going to change anything meaningful. If you do make meaningful changes, often those changes are negative. Only once in a while do mutagenesis actually produce something beneficial to the plant. What this meant was that scientists had to expose seeds, thousands and thousands and thousands of seeds to these mutagens, and then actually plant them and see what they got. Again, planting thousands of plants, going through, identifying what possible mutations occurred. More often than not, nothing significant changed. When the things did change, the plants often had negative mutations, but every once in a while, you got some sort of beneficial, pretty much randomized mutation that came out of this. Mutagenesis is basically like playing roulette with mutation, hoping you're going to get what you want out of it. It took a lot of work and many years, but many products, all the grains that we eat today, the modern grains that are farmed, much of the modern produce, have all been developed using some sort of mutagenesis technique. And we've gotten from that more resistant grains, crops that can hold up to drought, all sorts of benefits that we've gotten from mutagenesis over the years. One of the most common, or the stranger ones, I guess you could say, is a grapefruit. If this is what you think of when you think of grapefruit, this is the process of mutagenesis. Before the 1930s, grapefruit wasn't red, it wasn't pink. All grapefruit was white. These white grapefruit quickly fell out of favor, and I think now in most grocery stores, all you can find is red or the ruby red, pink grapefruit. Um, but yeah, the process of mutagenesis. Where mutagenesis was something of a crapshoot, editing DNA with this process called CRISPR, that's the way it's pronounced, CRISPR, but pronounced CRISPR. This is like going in with tweezers or a surgical knife and being able to specifically edit parts of DNA and the traits in DNA. I'm not going to try and create a video here that goes into the details of how CRISPR works. There are so many better examples out there on the internet, several of which we'll link to in the research itself and also in class and on Google Classroom. But the basic idea of CRISPR is that you're going in and you're able to snip out particular sections of DNA, and then you could either snip them out and keep them out, you could snip them out and replace something else in its place. You can make all sorts of very specific changes in the DNA to hopefully get what you want as a result. CRISPR has the ability to remove that randomness of mutagenesis, where instead of just looking for some sort of random positive outcome of a mutation, here we can choose what the outcome is that we want. We can make that change and then see that change through. So just a couple of quick examples of where we've used CRISPR in agriculture. You may be familiar with citronella candles used to ward off insects like bugs. You light the candle when you go camping or something. We have actually edited the production of those oils. It's the scented oils that repel the insects. And we have put those genes, those traits, into tomatoes so that the stalks of the tomato plant produce that same type of oil. Hence, the tomato then produces its own insect repellent so that they don't need to be sprayed with other pesticides. There was a breed of cow that, when on crowded farms, its long and pointy horns would often injure other cows. So they have engineered the horns out of the cow. They edited it so that the cow no longer grows horns. So this way you no longer have the horns bumping into each other. Some of you may be familiar with the fact that spider silk, that's the spider web, is extremely strong and durable given its very low weight and size. We've been using spider silk to weave things like bulletproof vests and super strong fabrics. The problem is that milking a spider takes forever. Basically, you have to pull the strand out one thread at a time. They have engineered goats to produce the same proteins used to make the spider silk, but the goats produce it in their milk. So we've made these spider goats. They look just like regular goats. They're normal goats, but when they're milked, the milk actually contains the base material for making the spider web. They're then able to separate that from the milk and use that material to make something, make all sorts of new materials. Like I said, really high tensile strength ropes and all the whatnot. Um, Doing this from an engineered goat. And then we've done some more crazy things like make featherless chickens. This way you don't have to worry about plucking the feathers off the chicken before you process it. Or a regular favorite, glow-in-the-dark cats. These cats, in fact, glow under UV light. Kind of like if you've seen when they turn on the black lights and neon kind of lights up and glows. These cats will glow under that as well. The point is, the possibilities are endless when it comes into using CRISPR for gene editing. The real question then is, where do we go from here? Do we continue producing enough food to feed the billions of people on this planet? Well, that sounds like an obvious direction to go in. Should we continue curtailing to some kind of odd pet market where we have glow in the dark cats and already plucked chickens? Where should we draw these lines? What changes should we make? What changes shouldn't we make? Those are questions that need to still be answered. So the next question is, where are we going from here? As a class, we are going to be having some discussions about what directions do we think should food production be going in. Should we continue using CRISPR? Should we put regulations on CRISPR? You've heard a lot of stuff about using and consuming genetically modified food, so the GMOs, genetically modified organisms. What's appropriate to be putting in your body? What's appropriate to be burning in the tanks of our cars? The point is, we need to kind of work our way through this to understand what directions and what steps humankind should be making in the near future. That being said, we're going to be introducing a new model in class, one that is used to help you build arguments. As your teachers, it's not our position to tell you what your arguments should be, so we're not going to tell you whether you should support using GMOs and CRISPR used in food production or whether you should oppose it. But you're going to take positions and you're going to have to defend those positions using evidence and fact-based research. More details will come on this later.